In this paper, we perform a gain-of-function screen to identify regulators of cell fate in the mammary gland, and specifically to identify transcription factors that enable luminal epithelial cells to adopt a basal-like fate. This question is potentially important to the understanding of how breast cancer heterogeneity is generated, since recent evidence suggests that basal-like tumors are likely derived from a luminal progenitor cell of origin. Our screen was based on previous findings that luminal and basal cells exhibit different phenotypes in culture. Namely, basal cells readily generate adherent colonies, whereas luminal cells lack this ability. Therefore, adherent colony formation could be used as a surrogate for basal differentiation. We first isolated mammary epithelial cells from primary tissues using a combination of enzymatic and mechanical dissociation. We then used cell surface markers and FACS to isolate the luminal subpopulation, and then we stably transduced these cells with a library of human transcription factors and lentiviral vectors. Finally, we harvested all of the adherent colonies that formed and recovered those transcription factors from the genomic DNA of the adherent colonies by PCR and high-throughput sequencing. For each transcription factor in the library, we derived an enrichment score using cells that were transduced but not screened as a control. And this approach led to the identification of 54 significantly enriched hits, which are listed in Panel B. Panel C is a scatter plot that shows the number of sequencing reads that were obtained for each transcription factor against that of the control sample. We wanted to go back and study some of these factors in more detail. We've started to follow up on several of these transcription factors in the lab, but the one that we chose to focus on initially was WWTR1, or TAZ, a key component of the Hippo pathway. We validated that TAZ cDNA alone could promote colony formation in luminal cells. The rest of the paper is focused on defining the precise role of this transcriptional regulator in mammary epithelial development and differentiation.